Okay, so over the last several chapters, and I don't know if you looked at this or not, but maybe the first page of the textbook, it shows you which authors wrote each chapter, and the same author wrote chapters 8 through 14. And so that's why there are a lot of similarities over the last several weeks. Over the last several weeks, we've talked about systems biology quite a bit. The author likes to say systems biologic approaches. And I don't know exactly what that means, other than that it's got systems biology at kind of in the background of explaining a lot of things. So chapter 14 does a wonderful thing, is that it takes a break from introducing new concepts to explain what systems biology is. And the reason why I say it's not introducing a new concept is it's something that we've referenced in several chapters but never explained. Okay? So it's like, okay, well, we have an idea of what we mean by systems biology. We're talking about something that it's not just genetics, it's not just physiology, it's not just anatomy, it's not just molecular biology, it's the study of biological systems, right? And so we have kind of this understanding, but we need to spend a little bit of time of explaining the uses and the intricacies of how it works. And that's what chapter 14 is all about. And it's fun. It's a fun chapter. I think you'll like it. I don't know you. Okay. So key terms. First one, systems biology. Great time to define this term. And so what this is a study of components and how they interact in living systems. So it's a study of, let's say, biological components and how they interact in living systems. So you all are familiar with the hierarchy of life, right? So it's like we start at the molecule, and then we go to organelle, and then we get to the smallest unit of life, the cell, and then we get to tissues and to organs and to organ systems, and then the entire organism. And we have this habit to make whatever we're studying at the moment the most important aspect, right? And so this becomes especially true the more knowledge you accumulate. So for those of you that go on and take molecular genetics and then go and study genetics in grad school, which some of you will, you'll have this habit of thinking, man, the genetics is the most important aspect of what makes an organism an organism. And it's really sad to think that way because of course it isn't. And you know from this class, which will get lost in some of the molecular genetics that you'll do, but you know from this class that there's a lot that happens in between the genotype and the phenotype, right? There's a lot. There's a lot that happens in between those two to determine what the phenotype is. And so, of course, genetics isn't the only thing, and it very well is probably not even the main thing. But anyways, we have a tendency to do that. The nice thing about systems biology is it is deliberately multidisciplinary. Systems biology is deliberately multidisciplinary. Meaning you cannot have systems biology without geneticists and physiologists. Physiologists? It sounds weird, but it works. And without molecular biologists, and without statisticians, and without anatomists, and without organismic biologists. Like you can't have systems biology. And the beauty of this is we're getting really, really good at modeling biological systems. We're getting really good at it. And with that comes the need to interpret what we see. And with that comes the need to build collaborative networks. And so what you're seeing in a lot of funding now, here, I'll tell you this. It used to be that if you wanted to get a big NIH grant or a big grant from the NSF, this big government grant that's a multi-million-dollar, multiple-year grant, it used to be very easy for you to, as an individual with a good publication record and a really good idea, submit and get that kind of funding for just your lab. That is almost unheard of now. For you to get access to multi-million-dollar, multiple-year grants, you almost always have to apply as a collaborative network. And oftentimes at multiple institutions or multiple labs within the same institution. And that the reason for that is because we're starting to see the value and the importance of systems biology. To not look at things as individual units or individual components, but to look at things as an entire system. Because it seems absurd to have like a mechanic that all they do is work on radiators, right? You're like, I don't understand this analogy. I don't really understand this analogy either, but I know what a radiator does. I mean, that's absurd, right? And you're like, well, we have engineers that design very specific parts. That's true. But when you are working at how do I get this entire system to function properly, and that is an automobile, like you don't really have specialists, right? You have people that are generalists. They may specialize in a specific type of vehicle, but they are generalists because the reality is every single component interacts with multiple components. Not every other component, but multiple components. No component is an island. No protein is an island. No gene is an island. No individual piece of mRNA is an island. It all interacts with other components. So listen.
Sorry, the system you're referring to, the biggest thing we're studying is the organism.
Yes.
Okay.
Yeah, if you want to start doing things above that and representing them as systems, that becomes very complicated. Very complicated. There are some efforts to do that.
Biology looks at the organism as a whole.
Yes. Mm-hmm. Yep. All right. Took me 10 minutes to explain that term. Okay. Next term, graphs. You're like, I know what a graph is. But a graph in systems biology, it's a little bit different. It's basically a map of interactions. That's what a graph is in systems biology. It is a map of interactions. Probably should be called a map and not a graph. Because we have a specific image in our head that we see when we hear graph, right? And it's like that XY axis and and if you're anything like me, you've got a nice parabola, right? But you've got a parabola that doesn't even hit the x-axis. It's all over the y-axis, so there are no zeros. You're like, wow, this is a really interesting parabola. Anyways, but so it probably be better again to refer to these as maps, okay? They are a map of interactions, these graphs in systems biology. And these maps of interactions contain basically two components. The first one is called a node. And so what a node is basically a point on your map, and it represents some biological component. That node could be a gene. That node could be a transcript. That node could be a protein. That node could be some kind of fully formed and functional RNA. Like maybe that node is a specific tRNA. So it's not just a transcript, but it's folded properly and fully functional. It's even charged, right? It's got that amino acid on it ready to go. Now, most often when we build these maps or these graphs, our nodes represent proteins primarily. Typically, our nodes are representing proteins. But because this is systems biology and we understand that proteins are not islands, they are influenced by other proteins and they are influenced by genes that don't get translated into proteins, those components are more than just proteins. But many of our components are going to be proteins. About 2 million of them in the human proteome. About 2 million different proteins. Now, edges. So again, these graphs or these maps contain two components, nodes, which are the components, and then edges. And edges represent the interactions among nodes. Oh my goodness. There's so many markers. It's like somebody was having a review session in here. And so here's what one of these maps might look like. So here are our nodes. And these nodes, again, they are representing biological components. They might represent a protein or a specific gene or a specific transcript. And then these right here are the edges. So here's an edge, here's a node, and we know that a node represents a component. like a biological component, and an edge represents an interaction. And so you can see that these maps or graphs, whatever you want to call them, they can become very sophisticated very quickly. Right? If you're like, okay, well, there are 2 million proteins in the human proteome. On top of that, there are hundreds of millions of transcripts. And then on top of that, there are 20,000 coding genes. Right? And you're like, man, this could get really complicated. And you're right. And so what we're doing with systems biology is not trying to represent the entire organism. We're trying to represent something that we've not really ever done before, and that's what is happening at multiple levels on the hierarchy of life to predict the incidence or prevalence of a particular phenotype. Okay? And so what we might do is we might build a graph or a map for a specific genetic illness, right? Like we might develop this map for, say, type 2 diabetes. And then what we would do is include as many of the components as we can that are going to influence this and as many of the interactions as we can. And so that way we can start to model this and make predictions on what would happen if. What would happen if? Okay. So I'm going to introduce this term. I'm not going to, it's actually 2 terms, but I'm not going to mention much about it right now because I have basically a whole slide on it. Scale-free networks and hubs. And just a little bit about this. This is a view of how to build these maps that suggest that there are going to be some nodes that are more important than others. Okay, so it's a strategy to build these maps or graphs in such a way that it notes that there are certain nodes that are more important than others. And if this is what we're doing, more important than others are going to be nodes that have more edges. Okay, they're going to be nodes that have interactions with more nodes. Does that make sense? Okay, we'll talk more about that in a little bit, so we're going to move on. The interactome. And so probably my favorite aspect of genomics is all the ohms that come as a result of genomics. So originally it was just the genome, and then it's like, well, then we've got the transcriptome, right, all of the transcripts that are made. Then we've got the proteome, all of the proteins that are made from this process of gene expression, and the interactome. And so the human interactome is the representation of all interactions between or among all components in the human system. And you're like, that's crazy. That's even bigger than a genome, and you're right. And so we typically, again, we don't try to visualize or work with the entire interactome. We work with pieces of it. Okay, we work with pieces of it, and oftentimes what we'll do is we'll work with specific phenotypes, specific characters. Maybe a genetic illness, maybe redheadedness. You're like, isn't redheadedness a genetic illness? I don't know. Okay, this next one might be my favorite, the morbid maps. Morbid maps. So these are graphs that represent which nodes correlate with genetic illnesses. Okay, so these are maps or graphs that illustrate which nodes correlate with genetic illnesses. And I'll show you a picture of 2 different types of morbid maps. Yeah.
Don't all nodes correlate with just like if any one of them is in the wrong spot, you're
Yeah, but we have to do this kind of piece by piece.
Oh.
Yeah, I mean, sure, if you're going to consider the entire interactome, then sure, anytime you mess with a node or an edge, there's going to be some corresponding genetic illness. But you have to keep in mind that when we build these graphs or we build these maps, we're dealing with typically a single carrier.
Okay.
Yeah. So like we might build a map for type 2 diabetes. And then we're talking about, okay, well, which nodes in that map correspond to the development of that pathology?
To a specific genetic illness then?
Yes, typically. Yeah. Perturbation. Perturbation is a modification of a interactome. Okay, so a perturbation, it's a modification or an alteration of an interactome. Okay, so you're messing with a node or you're messing with an edge. Okay, and we'll talk about both and then put those in the context of some of the concepts we've learned so far. And then, finally, diseasome. This is an interactive map that shows the relationship among genetic illnesses. So, we know this, that several genetic illnesses might be caused by the same issue, right? We saw this with our CYP genes. Right? When you have an inability to properly metabolize something, you can have several different physiological implications of it. We also know that there's what we call genetic heterogeneity. And somebody remind me what that term means. Genetic heterogeneity. So the previous example I gave you was basically one mutation, multiple phenotypic impacts. Genetic heterogeneity is in some ways the opposite.
So multiple mutations that all correspond to one
phenotype. Yep. So it's like one specific phenotype that is caused by many different mutations, and oftentimes many different mutations at different loci. And so all of our complex genetic illnesses are genetically heterogeneous, okay? In that all of our genetic illnesses can be triggered and influenced by mutations at several different loci, okay? And so what this aims to do, the diseasome and the interactome and just systems biology in general, it aims to basically model and better understand genetic heterogeneity, okay? So we're modeling all the different loci, all the different nodes that are involved in that particular genetic illness. Yeah, Kayla.
What is the difference between or the relationship between a diseasome and a morbid map?
So a morbid map is typically going to be done for a single genetic illness.
Okay.
So like you might develop a morbid map for type 2 diabetes.
Okay.
And so what it would do is you'd in your map, you would know all of the nodes that are involved in whatever it is that you're looking at. In this case, in type 2 diabetes, all of the various nodes that you can, that in some way are involved in the physiological process of regulating blood sugar levels, right? And then what you would do is you would, in these morbid maps, you would note which of those nodes have we found an error in that correlates with increased prevalence of the disease or increased propensity for the disease. And that's what we do with a morbid map. Okay, it's basically dealing it with a single genetic illness at a time. These diseasomes are modeling how does one disease influence another disease. Yeah. Okay, that's it for our key terms. So if we don't have any questions, we shall move forward. Okay? So here's the first question we need to talk about. How do we model the interactome? How do we actually do this? I mean, you know a little bit already. We're going to use these graphs or these maps. And so this is exactly what we do. We develop graphs that have nodes. And again, these are the interacting components. Many of them are going to be proteins. Some of them are going to be transcripts. Some of them are going to be individual genes. And we develop graphs with these nodes and also edges, that these edges are showing these interactions. Now, this is something ecologists have been doing for 50 years, maybe longer, using maps or graphs like this to model ecosystems, right? We call these food webs. And even you can do it not just in food webs, you can do interaction maps that are taking into account more than just feeding relationships, right? So are taking in like competitive relationships, mutualistic relationships, all sorts of different things. So ecologists have been doing this for 50 years. Biologists have been doing this for about 20 years now. Okay? They're getting on the train, and it's really starting to pick up some speed, to use, the train analogy. And so we have two strategies by which we can develop these maps. One that is good and one that is really good. Okay? We've got two strategies. One that is good and one that is really good. You're like, why wouldn't we just do the really good? I don't know. Okay, when these first started, we had what I would call the good. Okay, the good way to map the interactome. And it was done using what we call Poisson degree distributions. And so what these assumed was that pretty much, for the most part, nodes have the same number of interactions. Okay, for the most part, nodes have the same number of edges that attach to that node. And so we're looking over here. Oh, we need to do this. Okay. And now we're like, okay, we've got 1, 2, 3, 4 nodes, and each of these nodes has 3 interactions. And we're like, cool. And this is what we would use to build these, and we would use these to predict there's some interaction between these nodes that we don't even know yet, but we're pretty sure it's going to be there. Because again, we assume that nodes pretty much have the same number of interactions. And that having some nodes with very few or with more than the average, those are exceptions to the rule. Those are rare. Right? It'd be rare to find a node, say, like this node here, where it only has one interaction or one edge that touches that node. And we would say under our Poisson distributions, we would say these kinds of nodes are very rare. These kinds of nodes are very rare. Okay? And so that's one strategy. So again, a lot of these were used to predict interactions that nobody had discovered yet, and then you predicted that interaction would be in place, and guess what? You find it, and you're like, cool. And then we have what I think is the really good way. And these are called scale-free networks. And so in these networks, we don't assume any number of interactions. We basically assume that any node can make any number of interactions. That there aren't going to be some nodes or very few nodes that alter from the typical, and that almost all of them have the typical number of interactions. We're just like, you know what? A node could have anywhere from 1 to 5 million interactions, maybe even more. And what we're going to do instead, we're not going to assume the number of interactions. We're just going to model what we know, and we're going to instead use these to predict which nodes are what you might call essential nodes. Because while it doesn't assume that there's a set number of interactions for each node, it does assume that there are going to be some nodes that we'll call hubs that have a disproportionately large number of interactions. Okay? So this assumes that there are some nodes that we would call hubs that have a disproportionately large number of interactions. That they are involved in a number of influencing a number of other nodes. Okay, and we would call these hubs or essential nodes. Yeah, Kaylee.
An example would be like a Hox gene.
Sure. Okay. Yep. So, or like a initiation factor for transcription, or transcription factor. That's something more generally, like something like a transcription factor, where it's going to regulate the expression of a number of other genes, and we're going to assume that because we know that that's true from studying how development works, right? And so what we're going to instead do is we're going to build these graphs in such a way that we model all the interactions that we know, and then instead of predicting having interactions that we haven't discovered yet, instead what we're going to do is we're going to use these to predict nodes that, if they're altered in any way, are almost always going to result in some kind of a genetic illness. Okay. Yeah. Any questions about this? Yeah.
I can't imagine a gene that you could alter that wouldn't result in genetic illness.
Well, that wouldn't result. It's not really.
Like a phenotypic.
Yeah, like in something that's really devastating,
Okay.
right? And I mean, a lot of times you could have a mutation in a node that, as long as you have one wild type copy, you'd have no phenotypic, results, right? But then there are some of these nodes that are so integrated with other nodes that even having one mutant copy is enough to throw the whole system off. Okay, because then you've got a dosage issue. All right, so next question. How do our genetic illnesses behave inside this interactome? So when we say, okay, we're going to start applying this interactome to genetic illnesses, how do they behave inside of this? This is a wonderful diagram, came right out of the textbook. And so what this does is represent what we call the essential genes. And so far, this is saying about 25,000 human genes. It's actually closer to 21,000. But 1,565 essential genes, meaning if these genes are not in place, okay, and you're not making those protein products, life doesn't work, okay? And then we have disease genes, where a mutation in this causes a particular disease. Not a mutation here leads to a higher probability of developing the disease, but no, a mutation there causes a disease. We call these disease genes, and then there's an area of overlap.
So they correlate with monogenetic illnesses.
Yes, so this area of overlap is where you have an essential gene that correlates with a specific genetic illness. And you're like, well, what about all of these? Because you're like, a mutation here, of course it's going to correlate to a genetic illness. These are all areas where a mutation here, you don't get a living individual. The individual doesn't develop properly. The individual never exists past sometimes past even the zygote stage.
Wait, so
right?
what's the blue one, the disease genes?
Disease genes, yep. And
What
then,
does that mean?
so it's a disease that a mutation in there causes a specific genetic illness. Okay, so it's not a mutation there correlates with a higher probability of developing a genetic illness, but no, a mutation here causes a genetic illness. So a good example here would be the CFTR gene, where a mutation in that gene causes cystic fibrosis. Okay.
And the brown?
The brown is the area of overlap. Okay, where we have genes that are essential, necessary for life, but mutations actually lead to genetic illnesses. So you should predict from this area of overlap is probably in genes where they're essential to life, but where you start to see the phenotypic expression of them is later in life. Okay? Later in life, so that you can survive to a certain point before you start to see some of these genetic illnesses. All right. Now, most monogenic genetic illnesses remove nodes from what we call the periphery. So, if you're going to remove a node, what are you doing? What are these nodes representing?
Proteins?
They represent specific biological components. Sometimes it's a protein, sometimes it's a transcript, sometimes it could be an individual gene. But if you're going to actually remove a node, you have completely eliminated that protein. And cystic fibrosis, again, would be a wonderful example of this. Okay, cystic fibrosis, where you remove a node, that CFTR product, and it is necessary for life, and that's why you have eventually, at least in our current medical practice, eventually you are going to die from cystic fibrosis. You may live long enough to die from something else, but eventually you're going to die from that, because it is a mutation in an essential gene. And so this would be a monogenic genetic illness that actually removed a node from what we would call the core. Removed a node from the core, or the essential genes, one that has a lot of interactions. Most of our monogenic genetic illnesses remove nodes, so eliminate a product, but from the periphery, from where we have a node that is not connected to a lot of other nodes. And so it corresponds to one specific phenotype. So another good example of that, because cystic fibrosis is not a good example of the periphery. It is a good example of removing a node, but a good example of this would be PKU. Okay, PKU, phenylketonuria, in which you're missing the enzyme that converts phenylalanine into tyrosine. And so that would be, here you have removed a node. You've removed one of those biological components, but it is not one that is necessary for life. Life functions just fine without you converting phenylalanine into tyrosine. You can get tyrosine in your diet. There are other ways to make tyrosine. You can still have properly functional, tyrosine works a lot in cell receptors. You have what are called receptor tyrosine kinases. Anyways, but that would be one where you've removed a node and you've removed it at the periphery, okay? And that's where most of our monogenic genetic illnesses operate. They operate out at the periphery. They operate out at nodes like this, okay? That might have one interaction, okay? And that should make sense to you, because if you're removing nodes, you better not be removing nodes from the core, from those hubs, right? Because if you do that, it's going to likely be catastrophic. Now, most of our complex genetic illnesses, very few of these complex genetic illnesses involve removing nodes. Most of them instead involve modifying edges. Most of them instead involve modifying edges, changing the way these nodes interact. And most of them happen between hubs, in that core, in those nodes that are related to a number of other nodes, in those what you might call essential nodes or hubs. Okay, and so our different genetic illnesses operate in different ways and in different places. Does that make sense? Because if you're going to remove a node, that is going to cause a genetic illness. If you're going to modify how that node relates to other nodes, it may or may not cause a genetic illness, and you could probably overcome it by environmental factors. And we know this is true of complex genetic illnesses, right? We might have a mutation that correlates with a significant increase in the prevalence of type 2 diabetes. But it's like, you know what? You don't stress your system, you're probably not going to develop type 2 diabetes, right? Eat a well-balanced diet, exercise a lot, and even though you might have an incredibly increased propensity towards developing it, it's a pretty good chance you never will. But if you're American, you're going to develop type 2 diabetes. It's just how it works. If you live long enough and you're American, you're going to die of heart disease or type 2 diabetes. Or kids? Oh, cancer. I thought you said or kids. I was like, wow. Yeah, Caitlin.
So is that the case because if you modify an edge, you're only really modifying an interaction between a couple nodes? Whereas
yes.
if you modify a node by removing it, it interacts with all
absolutely.
The edges.
Yep, absolutely. Yeah, that's an excellent point. So if we modify this edge, we're just going to do this. This is going to be a wonderful visual representation for you. You're going to like this. Look at this. Boom, we changed that edge, right? We've modified it. It used to be black, now it's blue. Okay? And you're like, what we've done is we've changed basically one interaction. But if instead we remove this node, we've lost 3 interactions there. They're gone. Absolutely gone. And if this is going to happen at a hub, this is going to be devastating, like cystic fibrosis. Yes?
Can nodes also be environmental factors?
Yeah, but then we're going to build some really complex maps. But sure. Yep. Yeah.
So we can make a map of anything we want.
Oh, absolutely. And we should as systems biologists. All right, so I want to show you a couple of other illustrations. And so here is how these maps or these graphs typically look. Where here we've got our center, or what we might call our hubs. Okay, these are nodes that have interactions with many other nodes. And then here, and then these in red, these are the hubs. Okay, and this is like 1, 2, 3, 4, 5, 6 different interactions. 1, 2, 3, 4, 5, 6 different interactions. 1, 2, 3, 4, 5. That one only has 5. So sad. 1, 2, 3, 4, 5. That one only has 5 as well. But these are our hubs. These are our nodes that are essential nodes. If you remove these, very likely that it's just not going to work. Okay, that life will not continue past the zygote stage. Okay, or not very long after the zygote stage. And then here we've got our periphery. Okay, where our nodes out here at the periphery have very few interactions. And this is where, again, we're going to see most of our monogenic genetic illnesses are going to remove nodes out here in the periphery. And the blue here, these just correspond to nodes that actually correlate with specific genetic illnesses. Okay? And these are monogenic genetic illnesses. You remove those nodes, you get that genetic illness. Okay? And then our complex genetic illnesses are most of the time going to modify edges here. And that's what this is trying to represent. This, a disease module, or what you might call a morbid map. Okay, this is a morbid map or a disease module where it's showing you basically in red all of the nodes that if you modify these nodes are going to correlate with the genetic illness. You can't remove them. If you remove them, it's likely it's not going to work. But then also note some of these edges are in red as well, where we're mapping edges that if they're altered are going to correspond to an increased likelihood of developing a genetic illness. Alyssa.
What makes something red versus white? Because some of the white ones have more interactions with the red ones.
The specific genetic illness you're talking about.
Oh,
Yeah. Yeah, so we're gonna build this disease module or this morbid map for a very specific genetic illness.
Gotcha.
Yeah, not just any genetic illness in general. Because like you said, if we modify a node, in all likelihood, there's going to be some interaction for that. Now, the nice thing is there is some redundancy to protect against this. Okay. Yeah.
This is like relating to the previous slide, but I was wondering, for the first strategy, like what led to their thinking to decide that like.
Previous slide.
Yeah.
Well, that was just the first way we ever did.
To just assume that there was just the same set amount.
Yeah, I mean, in many ways, it came from the same assumption that was behind us predicting that there were going to be a lot of genes in the human genome. That the bulk of the genome exists to code for proteins. We had very little understanding of gene expression, like regulation of gene expression. And so if you don't have a good understanding of there being some portions of the genome that are going to impact other portions of the genome in more ways than just translating into some protein, then what you would assume is, okay, well, one gene gives us one RNA, gives us one protein. And every one protein has its job, right? And so you'd assume that basically every protein has the same number or the same weight in everything else. Now, within that, you would, I mean, if you were a good evolutionary biologist and you saw that there were some genes that were conserved from one organism to another, you would assume that those are essential genes, right? Why do you find the same molecular machine in bacteria, eukaryotes, and archaeans? Well, it's because it's necessary for life. So you would assume that that's very rare, right? And again, that those nodes that have a lot of interactions, those are rare. But it's just not the case, right? Because nodes that are regulatory are going to have a lot of interactions. We're going to have a lot of them. We're going to have a lot of nodes in that center. All right. Oh, why did this... Wow, sorry, that slide snuck in there a second time. I just skipped right past it. I don't know how it got in there a second time. So, same question, but going to a little bit of a different point. Perturbations, which anytime you change a node or an edge, again, that's going to be a perturbation. These perturbations from mutations, they're going to alter the expression of genes. We know this, right? We've seen this time and time again, where we've got regulatory genes, and so mutations are going to alter the expression of other genes. And we're noting that in this systems biology, because we're noting that nodes are connected to other nodes. And these change networks do a really good job of explaining to us the difference between the genotype and the phenotype. Because with monogenic illnesses, genotype basically equals phenotype, right? So like with cystic fibrosis, your genotype equals phenotype. Right? If you are a double mutant, you have cystic fibrosis. With these complex genetic illnesses, it doesn't work that way. Right? There's a relationship between genotype and phenotype, but it's not if genotype, then phenotype. Right? It's a little bit more complicated than that. And so what this does and what systems biology does in building these networks, these maps, these graphs, helps us to explain the connection between the genotype and the phenotype in a way that just doing genome-wide association studies can't do. In a way that just doing physiological studies can't do. And so here, this is a wonderful diagram right out of the text where you can see this node has been removed. You see that? From here to here, that node has been removed. And then what this will do is help us to explain how we get from here, our specific programming, to here, the specific expression of our development, of our genetics. And so what this diagram, I think, does a really good job of explaining is it shows us visually how we can explain genetic heterogeneity. How can we explain genetic heterogeneity? Well, what we do is we demonstrate all the different maps, all the different graphs that will result in the same phenotype, in that same genetic illness. So what we could do, let's use type 2 diabetes. I keep using this over and over and over again today. But with type 2 diabetes, we can generate several different maps, several different graphs that would all result in the same phenotype, okay, in the development of type 2 diabetes. And this is where it actually would be pretty helpful, Alyssa, to pull in some environmental factors as nodes in these maps, to build us several different really useful and helpful maps. Now, you probably wouldn't want to try to do it all on the same map, but several different maps that could all result in the same genetic illness.
So it's almost like if you were, not maybe you were missing one, but if one of your nodes was slightly moved, you could imagine like one of the environmental factors just like pushing it up.
Yes. Well, and then the opposite is also true, where you're like, if this node were absent, what is something we could put in its place to prevent the development of this genetic illness? And having these models, having these maps, having these graphs, modeling all the interactions helps you to do that, right? If this edge were gone, what could we do to replace it? Okay? That's it. It's 11:35. The quiz is now open. We got exactly